Hello and welcome to another one of D's ADD videos. We're going to be looking at vision, the big picture. As you can see, there's a lot of information that I'm going to cover here with the vision, but I'm going to try to break it up into chunks. So first we're going to look at the basic anatomy of the eyeball. How do we get all this information, visual information from the external world back to our brain? Well, it starts here. Light gets refracted by the cornea. Most of the light that gets refracted by the cornea and directs it through the pupil, the hole that goes through the lens. So the light basically takes a straight shot through the lens, and depending on how tight these ciliary muscles are, the lens will either be thin or plumper, and it'll direct the light back onto the retina here. And I'll go into the different layers of the retina in detail, but basically there are the layers that start with the ganglion neurons that go back to the optic disc and then end up at the outermost layer with the photoreceptors that actually receive the light and change it into an electrical signal that we can perceive with our brains. So you would think it's the other way around, and we'll go through that detail here in a little while. But that optic disc here is going to be the source of your blind spot, and the fovea is going to be your clearest vision. The reason why your fovea is your clearest vision, you have only cones, and cones are photoreceptors that respond to color light. And you might ask, what is a photoreceptor? Just in case you don't know, they're neural cells that respond to light. Okay, and the other type of photoreceptors present in the eyeball are rods, and they're responsible for black and white light. So that was rods with black and white light, cones with colors, C and C, if you want to remember it that way. However, you got to remember it. And the highest concentration of cones occurs in the fovea again. There are no rods in the fovea. And the highest concentration of rods occurs in the peripheral parts. Okay? And there are different types of ganglions that project. There's going to be your MP and we'll go through that in another video. So here is the basic layout of what's going on. I guess I can rotate it a little bit. Let's use the rotate feature. Oops. No, don't want that one. Sorry about that. Okay, rotate is messing me up a little bit. Okay, here we are. So we have a left visual field and a right visual field. And both eyeballs pick up both visual fields. Okay, so we're going to have the right eyeball. Notice that the left visual field shows up on the right side. It's inverted. And the right visual field shows up on the left side. Or the nasal portion, left, and temporal portion, right. And this is only for the right eyeball. And if you remember, the nasal portion will always cross over to the other side. That's called decussation whenever it crosses over. Decussation, or decussate. They go contralateral. And the temporal portion stays ipsilateral. Here we have the same scenario, but on the left eyeball. And notice that the right visual field is on the left part of the eyeball. And then we have the left visual field on the right part of the eyeball. Again, the nasal portion, in this case, the left visual field is going to cross over, whereas the right visual field will stay ipsilateral. Okay? And they are going to go to the LGN of your thalamus, lateral geniculate nucleus. And there are different layers of this nucleus in your thalamus. About 10% of your retinal neurons go to the superior colliculus, that's in the midbrain area. And these are responsible for the hot chick or hot guy response that you catch out of the peripheral of your eye, and then all of a sudden you move your eye to bring that into the center of your visual field. Or if you want to think about it in terms of nature, it's your, oh crap, I think I saw something move in the bushes to the right of my peripheral vision. And then you look, move your head, coordinate with your motor neurons to move your head in that direction, bring that region into your central visual field. Okay, that's the superior colliculus. All right, and it's found in the tectal area, the midbrain. And there are some neurons from the retina that also go to your suprachiasmatic nucleus that are responsible for your 24-hour light cycle. Some people cannot sleep because light hits their eyeballs, and there's a good reason behind that, and we'll discuss that later when we talk about the sleep cycle. Okay? And then finally what happens from there is we get optic radiations that go back to your occipital cortex, and there are 6 layers, inside out layers of the occipital cortex that help process your visual information. All right? So that's the big picture there of that one. Gotta rotate back to 0. Oops, wrong direction. There we are. So this is a photoreceptor membrane. So if we're looking at, this is a cross-section of the retina. Okay. We're going to have, these are your ganglion cells here. Oops, sorry. And we're just having a field day here. The ganglion cells project out to the chiasm, then to the different spots I just talked about. But if you look at the very back of the retina, we have these photoreceptors. Some are rods, some are cones. All right? And if you were to zoom in on one of these guys and take a little cross-section of the membrane, this is what you would have here. Okay? And what's going on is this is the way light's processed. And you have these players, opsin, retinol, G protein, and phosphodiesterase. And basically what happens here is there's a compound called cGMP, cyclic guanosine monophosphate. You can see why they call it cGMP, but it's basically a cyclical molecule that has a specific shape that fits into this channel and keeps it open in dark times. This is dark, okay? And then when the stimulus hits it, it activates the retinol, then the G protein, which wakes up the phosphodiesterase, which starts chopping up this cGMP. Okay, phosphodiesterase. Its role is to take cGMP, which is the key to open the sodium channel, and convert it to this, which no longer will open the sodium channel. So this is an evidence of a system that has 2 unique features. One, that light is what activates this channel, metabotropic receptor, the G protein receptor, which then kicks off the GDP here and makes it a GTP bind. And then the alpha subunit wakes up phosphodiesterase, which chops up the key that keeps this channel open, and the channel closes. Okay, so this is an example in which a stimulus actually shuts things down instead of turning things on. So here we have some a cross-section of your retina. Again, it's inside out. You would think that the photoreceptors are out here, but they're actually in the way back part of the retina. We have some rods and cones. Okay, we've already discussed those. Cones, color vision rods, are going to be the black and white vision. Here we have some horizontal cells, D, and what they help do is integrate the information between adjacent neurons. So F the horizontal cells and macron cells do the same thing. Responsible for phenomena like lateral inhibition. Okay, basically, when one stimulates, the adjacent ones can be inhibited. Okay, so now don't forget, whenever this is a stimulation, it's not a depolarization, it's a hyperpolarization. Why is it hyperpolarization? Because we have shut down this channel. And sodium coming in made it more positive, so if sodium doesn't come in anymore, it's going to become more negative. And then the neurons go back to the LGN S suprachiasmatic nucleus, lateral geniculate nucleus, or superior colliculus. Okay, so these are just some definitions you can write down if you want. I suggest making note cards for all this. This is a lot of information, but that's basically it in a nutshell. Make sure you check out the videos that have to do with the individual layers of the LGN and corresponding layers of the occipital cortex. Hope this has cleared up any fundamental confusion you might have about vision. Thank you and have a nice day.